So this morning I'm going to talk about rapid microbiological methods, and more specifically Legionella and Mycobacteria determination by quantitative polymerase chain reaction, or qPCR. Legionella is defined in ISO 11731 as a genus of gram-negative bacteria which will normally grow in not less than 2 days on BCY, which is Legionella-specific isolation agar, and capable of forming colonies which are often white, purple to blue, or lime green in colour. ISO 12869, which is the qPCR method, defines Legionella as a bacterial genus which can be defined by DNA sequences of genes which encode genus and species-specific 16S ribosomal RNA subunits. There are over 50 species of Legionella which we've discovered, of which at least 20 have been found to cause disease in humans. The most significant species of Legionella in terms of public health is Legionella pneumophila, but any Legionella species isolated is potentially pathogenic or should be treated as such. Mycobacterium, in this case we're not talking about Mycobacterium tuberculosis, we're talking about non-tuberculosis Mycobacterium, and they're very slow growing, they're non-spore forming, non-motile bacilli, which is rod-shaped bacteria, and these can take up to 28 days to grow by culture methods, whether you're growing them in agar or whether you're growing them in broth systems. They're relatively ubiquitous in water and soil in the environment, and they can accumulate in water systems under similar conditions to those where Legionella proliferates. And they pose a risk to similar populations at risk of infection from Legionella, although typically a non-tuberculous Mycobacterium infection isn't as immediately life-threatening as Legionnaire's disease. And over the past 2 years, they've emerged as a potential cause of post-operative infection in hospital operating theatres. So qPCR, which is the technique we're talking about today, is quantitative polymerase chain reaction, and it's a way of amplifying specific DNA present in a sample and determining how many copies were present originally. As one bacterial cell should have one copy of the relevant gene or DNA sequence, we can then count back from our amplified reading to the number of copies originally, and therefore deduce the number of cells present originally. With qPCR, the length of time the reaction takes to reach a certain number of DNA copies is how we do this, because every time we go around a PCR cycle, we double the number of copies present. And we will express our units, our results, as a genomic unit per litre, so 1 copies of DNA per litre. And this differs from culture, because in culture, one or sometimes more bacteria which land on the agar together will grow to form a colony, and we'll report those as colony-forming units per litre or per volume examined. In terms of what the results mean, qPCR counts the total number of bacterial cells present, and that will include non-viable cells or dead cells, which still have their DNA intact, and viable but non-culturable cells, as well as viable culturable cells, which you'd normally detect by the standard culture methods. Culture will count only the viable and culturable cells, and therefore it misses potentially disease-causing bacterial cells which you can't grow on agar in the laboratory. PHE has done quite a lot of work with this, particularly with Legionella, and what they've shown is that if your results by qPCR are below the reporting limit or the limit of detection for your method, you've got more than a 99.7% correlation with getting a negative result by culture. To put it more simply, if you can't find a Legionella or Mycobacteria DNA in your sample with qPCR, you're almost certainly not going to find any Legionella or Mycobacteria by the culture methods. But obviously, you can do this an awful lot quicker, which is where the benefit comes in. In both cases, culture methods, whether for Legionella or Mycobacteria, are still treated as the gold standard in industry. So, for example, for Legionella, if you're complying with L8 for Legionella control, you need to do culture to prove that your system's clear. And it's usually, but where it's really useful to use qPCR is as a tool for rapid investigation of systems, particularly critical systems, in the event of getting positive results by culture, whether your positive results by culture were derived from routine screening or investigating outbreaks. And because qPCR will give you your results within 24 hours, or sometimes quicker, it lets you target remedial action in a system much more precisely and much faster to eliminate the highest risk areas first, and hopefully to bring the area which has had to be shut down back into use sooner, which will obviously benefit a user of a facility because they're able to reopen it, but it also means that with the highest risk areas able to be treated first, you reduce the risk to users of the facility. If you're using qPCR as part of routine monitoring, it will also give early warning of potential of a system developing problems because it's more sensitive than culture methods and will detect bacteria earlier. If qPCR counts in genomic units start to increase, they can give early warning of potential problems and allow remediation to be considered or started before the system starts to show positive culture checks and potentially eliminating problems before they occur. The thing we're working on most in the future is we're making some modifications to the qPCR methods so that we're able to target, as well as measuring everything as we currently do, but we're also targeting measuring only viable cells. So this will still measure cells that are alive and have an intact cell wall, but which aren't able to grow in culture. Thermal control. So if you've got high temperatures, this can send particularly Legionella into a viable but non-culturable state, as can partial disinfection, where a chlorination has partly penetrated a biofilm and stressed organisms, but not actually killed them. These can then still recover and go on to reseed a system. And being able to measure this with qPCR is a potentially very useful application. We're still developing this and hope to have an announcement within the next 6 months that we're able to offer it commercially. In terms of collecting samples, we should take these in exactly the same way as a normal microbiological sample, and we should use a 1 litre sterile microbiological sample bottle. Commercially available ones are fine, but Lattice can supply them if required. Our email address is bottlerequests@latticescientific.co.uk, or you can contact us on 0208 853 3900 and we will arrange to have them sent out. We can deliver them to your office, to your site. We can also arrange courier pickup and delivery straight back to the laboratory to start analysis as soon as possible. If you're planning to sample for culture as well, then we would need to take a second sample bottle because we will use the whole litre of sample to process qPCR. And I would like now to give the opportunity to anyone on the call to ask any questions.
I'm okay.
Okay, if you do think of anything, please give us a call, and we'll be very happy to help.
Okay. If you can just let me know that the YouTube details so I can have a look at it on